Estoy acá para presentar a nuestro próximo disertante, que es el doctor Ben Hayes. El doctor Hayes es investigador principal en el Departamento de Industrias Primarias de Victoria, Australia, en el área de mejoramiento genético de ganado y especies de acuacultura. Se ha especializado en programas de mejora con información molecular y más recientemente en desarrollos bioinformáticos y análisis de secuencias. Doctorado en genética animal por la Central Queensland University. Ha dictado cursos sobre genómica, selección asistida por marcadores y bioinformática en Australia, Noruega, Canadá e Italia. Es autor de numerosas publicaciones, le pregunté recién, y son más de 70 en revistas de primer nivel, y capítulos de libros sobre mecanismos genéticos que controlan la expresión de rasgos cuantitativos, mapeo y selección asistida en ganado y especies acuícolas. Quiero destacar un trabajo de Ben, publicado en Genetics en el año 2001, de Meusen, Hayes y Goddard, sobre evaluación genómica, que marcó un antes y un después sobre este tema, y que pocos años más tarde fue adoptado para la evaluación. Fue un verdadero milestone, digamos, en el tema, y que seguramente Ben les contará a continuación.
So thank you very much for that introduction, and thank you to the organisers for inviting me to be here today, and for going out of their way to make my stay here very good. Okay, so the title of the presentation is Genomic Selection to Improve Production from Livestock and Crops. And I'm going to focus mainly on livestock here because Jose Crosser is going to give another talk on genomic selection in crops tomorrow night. So the focus here is mainly livestock with a bit of crops. So an outline of the talk. First of all, I'll be describing the challenge in front of us in agriculture and an opportunity that we have. And that opportunity is around genomic selection. So I'll be describing that technology. Then some new traits which this technology allows us to breed for, particularly in livestock, and then what the future looks like for this technology. So the challenge. The big challenge that all of us that work in agriculture really have is feeding about 9.5 billion people by 2050. How are we going to do this? So in crops, what does this mean? It means we have to improve the yields of crops, but at the same time, the resources to do this are going to be constrained. So we have to improve yields while improving water use efficiency and the efficiency of using inputs like nitrogen, for example. In livestock, all the predictions are that there's going to be increasing world demands for livestock products like meat and milk. However, while we can improve the production of meat and milk, the quantities of these, we also have to do this with these resource constraints. So we have to improve the efficiency of the use of feed, and particularly in ruminants, we have to do this while decreasing the amount of methane per unit product, at the same time as maintaining the value of the product. So that means maintaining high-value meat and milk products. So how are we going to meet this enormous challenge? Well, the good news is that animal and plant breeding have been helping us meet this challenge of feeding an increasing population for many, many years now. To give you some examples from animal breeding, the time to market weight for meat chickens has decreased from 16 to 5 weeks in just 30 years. That's a pretty dramatic increase. In Atlantic salmon, which has been farmed for far less time, the feed conversion efficiency has improved by 140% in just 7 generations. Again, a pretty spectacular increase in efficiency through animal breeding. In crops, the story is pretty much the same. There have been some very dramatic increases in yields over a long period of time through plant breeding technologies. So, for example, there was a paper published back in 2004 that said the genetic trend, the contribution of genetics to increasing yield, was about 77 kilograms per hectare per year, year in, year out. However, if we're going to meet that challenge of feeding 9.5 billion people by 2050, we have to go even faster than this in our genetic improvement. And how are we going to do this? And this is where the new genomic technologies come in. So if you've been tracking the DNA marker technology over the last 5 years, for example, you will have noticed a really dramatic decrease in the cost of getting one data point, the cost of getting a genotype at one DNA marker. And in the species I work with, the cost of the DNA markers has come down by about 1,000 times in the past 3 years. That's a very dramatic decrease there. And that's through this SNP chip technology. So this is these glass slides with many DNA probes on them. You can wash the DNA from an individual over those glass slides, and they'll tell you the DNA marker information at up to 800,000 DNA markers at once. So the challenge for us as animal and plant breeders is to use this technology, which is now being used very widely by the human geneticists, but to use this technology in animal and plant breeding. And the question is, can we use it to increase the rates of genetic gain? One way you could use the technology is through marker-assisted selection. What you would do if you were going to use marker-assisted selection would be to test each one of those markers in turn for an association with the trait you were interested in. Just say that was milk production in dairy cattle. You would test each of those 800,000 markers to see if one of them was associated with an increase in milk production. If you could find such a marker where one allele increased the production of milk or was associated with an increase in the production of milk, then you could use this marker in marker-assisted selection. Now, the benefit of using the markers in this way, these individual markers, depends on the proportion of the variance they explain in the trait that you're interested in. So let's have a look at a couple of traits to see how much variation individual markers explain. So the first trait I'll look at is a trait which is not economically important, but is of great interest to geneticists, or has been over the years, and that is a coat colour phenotype. So here the trait is proportion of black on coat. And you can see in Holstein Friesian dairy cattle, this ranges from anything almost completely black to almost completely white. Now, if you do a genome-wide association study, and in this case we tested 50,000 SNPs for an association with this trait, you do indeed find some markers that are very significantly associated with the trait. And here's an example on chromosome 6 in cattle. This is the position of the markers along the x-axis, and this is how significant it is as an f-value on the y-axis there. And in this region right here, there's a very significant association there. And it turns out that that marker is right on top of a gene called KIT, which is known to cause variation in coat colour phenotypes in many different mammalian species. So we've found a SNP which is associated with a gene affecting this proportion of black. The question then is, how much of the variation in this trait does this top SNP here actually explain? if we're going to do marker-assisted selection for this trait, how much extra benefit would we get from the marker? And it turns out that although the association is very significant, it only explains 9% of the phenotypic variation. This turns out to be a best-case scenario. If you look at traits that are economically important or important for production, like growth rates, milk production, the biggest SNP effects we find explain less than 1% of the phenotypic variation. In some cases, much less. The same seems to be true for some traits of interest in crop breeding as well. And this is an example from the classic Illinois maize or corn selection experiment, where they selected over a very long period of time for increased oil concentration in the maize kernel. And you can see this is the response to selection here over time. This is the percentage of oil in the kernel, just goes up and up and up over time. But when these researchers look to see how many genes explain this selection response, another way of saying that is how many genes underlie the variation in oil concentration, what they found was they actually needed a large number of genes to explain the genetic variation they were seeing. And in fact, they needed more than 50 sites along the genome to explain 50% of the variance. In other words, each one of those regions is explaining on average just 1% of the variance, and they're the biggest ones. So we've got a challenge here. We want to improve traits like yield, percent oil in the kernel, for example, but how do we do it when we have this genetic architecture where there's lots and lots of genes, lots and lots of mutations contributing to variation in our trait? Marker-assisted selection just isn't going to capture enough of the variation. An alternative is to use all the marker information simultaneously, or all the SNP information simultaneously. So rather than focus in on one part of the genome, just use all the markers spread across the genome simultaneously. And in this way, you can track all the chromosome segments, all the bits of the genome, and capture all the variants affecting this trait. And this is termed genomic selection. To show you how this works, what you need to start off with is a reference population. And in your reference population, you have many animals, in this case, or it could be many plants, that have both genotypes for many DNA markers across the genome, and they also have phenotypes, the trait of interest that you want to improve, like growth rate, milk production, and so on. What you can do once you have this reference population is calculate the effect of each marker. So here's just a cartoon of a chromosome here where we have markers, and in between the markers we have the effect of these mutations on our trait. And they might have a small effect or in fact a zero effect. And what we're going to do is estimate the effect of each of those markers, regardless of how many we have, on our trait. And in this case, we have one marker here. It's got two alleles. It's a SNP. It's got the alleles A and T. And we can work out from our reference population that if an animal gets a T allele instead of an A allele, it gets an extra 200 grams of growth. So the weight we would then put on that marker is 200 grams of growth. We're going to do that not just for a single marker, but for all the markers across our genome. If we've got 800,000 markers on our SNP chip, we're going to use 800,000 markers. And that's going to give us a prediction equation. And the prediction equation is just the effect of each of those markers by the second allele at the marker. So if you get a T allele, it's the effect of getting that T allele. Once you've got that prediction equation, once you've calculated that, you can go to any individuals in the population that don't have their own phenotypes, so they haven't been measured for the trait, but they do have genotypes for all the markers. And then you can just run their genotypes through the prediction equation, which sums up the effects over all these markers of the genotypes they have to get estimated breeding values, so the value of their genes. And then you can make selection decisions on that. So that's genomic selection. To show you how well this works in practice, I'll give you an example of something we recently did in Australian dairy cattle. So we're now applying this to select in Australian dairy cattle. The reference population we had was 3,800 Australian dairy bulls and about 9,900 cows. They've all been genotyped for 56,000 genome-wide SNPs. So this is markers spread right across the genome. The phenotypes, for the example that I'll use here, for cows, it's just how much milk they produce. Bulls, of course, you can't get a milk out of a bull. So for the bulls, it's the average of their daughter's milk production. Now, we have to have a way of assessing how well we're doing in predicting these genomic breeding values, predicting genetic merit. So what I'm going to do is split the data set into two. I'm going to have my reference population, which is in fact all the cows and the bulls born before 2003. And then I'm going to have a validation population of 274 bulls that are born after 2003. So what I'm going to do is work out the effect of each of those 56,000 SNPs on milk production in the reference population, then predict breeding values based on the markers for these 274 bulls. Then I'm going to calculate the accuracy of my prediction as the correlation between the genomic breeding values and their phenotypes, how good their phenotypes for milk production actually are in the 274 bulls. So how well do I do? The accuracy of the genomic breeding values, this is how well I can predict those, in this case, highly accurate phenotypes, is 0.75. Now, compare this, if you want something to compare this to what the bull breeders had before on these animals, which is the parent average accuracy. So this is the average of how good mum's genes are and how good dad's genes are. Just add them up and divide it by 2. That's the parent average accuracy. And that has an accuracy of only 0.35, so quite low. We're more than doubling the accuracy here. Now, what's the implications of this? To demonstrate to you the value of increasing the accuracy from 0.35 to 0.75, I'll show you how this has changed dairy cattle breeding programs in Australia and around the world, in fact. So this is what a dairy cattle breeding program looked like up until a few years ago. What the bull breeding companies would do is go out into herds around the country and look for bull calves with good parent average accuracy. So look for bull calves where the average of mum and dad's performance was pretty good. Now, the accuracy of those breeding values, selecting those bull calves, was pretty low. So what they would do then is a progeny test. They would take in about 100 bull calves, they would wait till they were reproductively able, then mate them to cows. You would have to wait till those cows had daughters born. Their daughters would grow up. You would start milking their daughters, and finally by year 5, when you're out here, you would finally get a performance record on these 100 bull calves. You would work out the value of the genes of these bull calves, their genes for milk production. And that's a progeny testing scheme. And it took 5 to 6 years to get those evaluations. Now, it's a very accurate way of selecting animals, very high accuracy, but the generation interval is very long, and it led to about 1% genetic gain per year for milk production. What you can do with genomics now, though, is because it's quite cheap to screen these bull calves for the genetic markers, you can screen thousands of bull calves on their genomic breeding value. And you can select them almost at birth for how good their genes are for a trait like milk production. And then you just have to wait till they grow up and are able to reproduce till you can use them in industry. So what you've done here is cut down the generation interval by 3 years or more. What effect does that have on genetic gain? The formula for genetic gain says genetic gain is the result of how intensely you select, how many bull calves you select out of how many there are, times the accuracy of selection, times the genetic variation we have for our trait, divided by the generation interval. What you're doing with this genomic selection scheme here is you're reducing the generation interval, this L, by 3 years. You're cutting it in half. And as a result, because you're dividing by 2 years instead of about 5, you're actually increasing the genetic gain by about 100%. You're doubling the rate of genetic gain. This has been an extremely attractive thing to do in dairy cattle breeding programs. This prospect of doubling the rate of genetic gain has led almost every dairy cattle breeding company in the world, I think, to adopt this technology and to start using it for selecting their bull calves to use out in the industry. So in dairy cattle, it's really taken off. In other industries, it's important to consider what traits are really important in your breeding goal. The opposite to dairy cattle, if you like, is something like meat sheep. So lamb, if you're running a lamb breeding program, lambs grow up extremely quickly. The main trait you're interested in is actually growth, how fast the lamb grows. You can measure that before the animal is able to reproduce. So there's not that opportunity to cut down the generation interval like there is in dairy cattle. So in meat sheep, there's not so much opportunity for selection here. Other species and other industries, though, for traits like female reproduction, where you get how good you get the value of a female's genes for reproduction after she's been selected to go into the breeding program, genomic selection is going to have quite an impact. And a range of authors have actually worked out how much impact this genomic selection technology could have on the breeding programs. So dairy cattle is an almost perfect place to apply it because you can cut down the generation interval. Meat sheep, you can get a small increase in the rate of gain because you can measure your traits or predict your traits more accurately. And in beef cattle, there's a big range here depending on what you're really selecting for. If you're going for very high value meat, so lots of marbling for example, then the potential for genomic selection is quite large. So that's the genomic selection technology. In the next section, I'm going to describe how we can apply this technology to new traits that we haven't really been able to select for in the past. And this gets back to this challenge of feeding 9.5 billion people by 2050. What are the traits that we really want to select for? And in livestock, one of the key traits to select for, if you think the supply of feed is going to be limiting, which it very likely is, then we have to improve the efficiency of converting feed into meat and milk. So we have to improve feed conversion efficiency. And in fact, there's a big profit motive in doing this as well. In dairy cattle, feed costs are anything between 40% and 70% of running the dairy farm. In beef production, it can potentially be an even larger cost. Now, we know there is genetic variation for this trait of feed conversion efficiency, but the problem in selecting for it in the past has always been it's very expensive to measure, because you have to measure how much feed an animal is actually eating, how much feed it's actually taking in. And that's an expensive thing to do. And in Australia, it costs about $800 to make that measurement. So it seems like a good trait for genomic selection. If we could have markers, genetic markers for this trait, then we could start selecting out in industry for feed conversion efficiency. Much cheaper than actually measuring individual animals for the trait. So there is quite a bit of work going on to try and get genetic markers for feed conversion efficiency. Just to give you an update on what we're doing, there's a large collaboration going on between Australia and New Zealand, where we have measured 2,000 dairy heifers for their feed conversion efficiency. This is how much feed goes into the heifer to produce how much growth. And we've also genotyped these heifers for 800,000 SNPs. Just to show you how much variation there is in this trait, which we've never been able to select for before, if you think about those 2,000 heifers and you take the 10% most efficient and 10% least efficient, there's a 1.5 kilograms per day difference in how much those animals are eating to grow at exactly the same amount. So in other words, all the heifers were growing at about 1.2 kilograms per day, but the most efficient were only eating 9 kilograms a day to get there, and the least efficient were eating 10.5 kilograms per day. So that's a big difference in efficiency there. But of course, you can only select on the genetic component here, and the heritability of that difference or that variation is 30%. So 30% of that variation is genetic that we can use in our selection programs. Because we genotyped all these animals in this trial, these 2,000 heifers, we can calculate genomic breeding values for this trait of feed conversion efficiency. And when we do this, we have an accuracy for this trait of about 0.4. So much lower than for milk production, where we have many records, but still a decent accuracy. And if we started selecting for that trait, feed conversion efficiency, the amount of feed that cattle would need to eat to produce meat and milk would actually go down, and it'd have quite an impact on the industry. So that's an example of how genomic selection can be applied to a new trait that hasn't been able to be selected for before. The last topic in the presentation is the future. What does the future of genomic selection look like? So two aims really, and they're both quite simple. The first is more accurate genomic breeding values. The more accurately we can predict the genetic merit of our animals and our plants from the DNA markers, the more rapid genetic progress we can make. The second aim for the future is to lower the costs of doing this. At the moment in the dairy industry, to genotype a bull costs about $90, I think, whereas to do the progeny testing scheme I described before costs about $30,000. So the genomic approach, genomic selection approach, is really beneficial to the dairy companies. It really cuts down their costs. For other species like meat sheep, for example, it isn't quite so attractive. But if we can reduce the costs of getting these genomic breeding values, we can really start applying this technology across a wider range of species. More accurate breeding values, just to touch on how we're going to get there. 2 ways to get there. One is larger reference populations to estimate the marker effects. The other is whole genome sequencing technology. So bigger reference populations, this is more individuals genotyped and phenotyped. What I have here is a graph of how accurate these genomic breeding values are given how many individuals you've got in your reference. So you can see there, if you've got 6,000 individuals in your reference and your heritability of your trait is 0.3, then you have an accuracy of about 0.5 over here. If you have a less heritable trait, so there's more environmental noise, you need much larger numbers to get decent accuracies of genomic breeding values. So the future is really about increasing the size of these reference populations. And one cost-effective way to do that, so you don't have to pay for it all yourself, is to get involved in international collaborations that are going on. And for example, in feed conversion efficiency, there's a large international collaboration which has about 10,000 animals involved in it at the moment. Okay, the other way of improving the accuracy of your genomic breeding values is through better DNA markers. So the current SNP chip technology, it's really fantastic, it's relatively cheap, relatively easy to use, but we do know that it only captures a proportion of the total genetic variance. In dairy cattle, we're capturing quite a lot of the genetic variance, 60 to 90%, depending on trait. But in some sheep populations, we're only capturing between 30 and 50% of the genetic variation. And this is the same problem that the human geneticists are calling the missing heritability problem. We have that in sheep and to a lesser extent in dairy cattle as well. And we also have the problem, because our markers aren't close enough to the mutations actually affecting our traits, we're getting poor prediction across breeds, which is quite a problem in something like beef cattle, where we have lots of breeds. So what are we going to do to improve this? Well, the ultimate DNA markers are in fact not DNA markers at all, but is the complete genome sequence of our individuals. So a few years ago, this was impossible even to contemplate because the cost of sequencing an entire genome was just prohibitive. When the human genome was released, the cost that was bandied around for doing that project was around 300 million, though some people say it was a lot more. In 2010, the cost was down to $5,000 for doing a complete genome. Now it's in the order of $2,000, I would say. However, that is the ultimate DNA markers, because then you have all the data, including the causative mutations. However, $2,000 is still too expensive to go out and sequence all the individuals that we need to make accurate genomic predictions. Remember that for most of our traits, the effects of the mutations on our traits are quite small. So we still need lots of animals or lots of individuals to estimate them accurately. In livestock and in some crops, I think, there's an alternative strategy we can use to sequencing all the individuals. What we can do in these populations is sequence the key ancestors of the population, and then use the fact that the chromosome segments from these key ancestors that we see in the current population trace back without much recombination to those key ancestors. So in the fairly recent past to those key ancestors. So we can impute that sequence data fairly accurately. And to give you some idea of how important these key ancestors are, these two bulls, Chief and Mark, between them they would have over hundreds of thousands of daughters. And in fact, this bull here, Mark, if you look at pretty much any dairy cow in Australia, 10% of her genome will be derived from this bull. And that's despite the fact that bull was from the US. So the strategy looks like this. If you can imagine those key ancestor genomes completely sequenced, then we have their descendants, which are the cows or the bulls in our population, and their genotype for these SNP arrays. So we just have a marker here, marker here, marker here. But because these key ancestors pass on their chromosomes and there's limited recombination, because there's not that many generations to chop them up, we can do a pretty good job of tracing back using these markers which chromosome chunks they've inherited. So then we can fill in the genome sequence data. And once we do that, we can do genomic selection on this imputed sequence data. Now, when you start thinking about this, you realise that for many of our cattle populations, our common ancestors are common across the world. And this is true in dairy cattle and beef cattle as well. So many groups are going to have the same key ancestors. So it makes sense to work on this in a global way, as an international consortium. So we've set up this 1,000 Bull Genomes project to put together the sequences, the whole genome sequences of these key ancestor bulls. And as I said, it's a global effort. We've got about 10 groups involved at the moment, but if you've got any whole genome sequence on any bulls, you can potentially become a partner in the project. And if you do, what you get back is all the sequence data from all the bulls in the project. So to give you an idea on where we're up to with this, we're up to run 2.0. We have 238 whole genome sequences from bulls of 4 breeds: Angus, Holstein, Jersey and Fleckvieh. When you put all that sequence data together, you detect an enormous number of DNA variants. So we're finding 25.3 million variants. I think that's actually more than the human geneticists find when they do a comparable number of humans. So massive diversity there. The next big challenge, of course, is predicting genomic breeding values using all that data. But that'll keep us busy for the next 3 or 4 years, I would say. If you are working in cattle and you're interested in becoming part of this project, you can go to this website and read about how to become part of the project. Okay, just finally, genomic breeding values. We've talked about more accurate genomic breeding values. Another big challenge is getting lower cost genomic breeding values, so it can be more widely adopted across our industries and adopted by more industries. Lower cost, there's already a way of doing this that I described, and that is to genotype individuals for lower density arrays and then impute up to higher density and calculate genomic breeding values from that. Another way of lowering cost, which is being explored very much in the plant world at the moment, not so much in the animal world, is to turn the power of these sequencing machines that just churn out enormous amounts of sequence data, instead of doing entire genomes, to do small chunks of genomes of many, many individuals at once. And that's called genotyping by sequencing. And that's another way of getting low-cost genomic breeding values. If you're interested in this technology, I think it's really worth, even if the current cost of the markers is too expensive, to start putting your reference populations together now. So what you need is individuals with good trait phenotypes for whatever you're trying to breed for, and something you can get DNA out of. So hair in the case of cattle and leaf or something in the case of plants. And if you start putting that together now, then you're one step ahead when the cost drops enough to get into it. So finally then, conclusions. To sum up what I've talked about, genomic selection can potentially double the rate of genetic gain in some species, where there's this opportunity to reduce your generation interval. So there's really the opportunity to increase your genetic gain. There's also this opportunity to select for new traits which contribute to efficiency that we haven't been able to access in the past. And feed conversion efficiency is an example of one of those. And genomic selection, I think, is going to be part of meeting this challenge of feeding 9.5 billion people in 2050. It's not the only solution, of course, but it is part of working out how we're going to get there. And it's rapidly becoming cheaper, so we can apply it across a wider range of species and industries. And if you want to apply it, start collecting your samples now. And finally, just some acknowledgements. I'd like to thank the bodies that fund us, and this is a list of my co-workers that produced some of the results that you saw here tonight. Thank you. Okay, that's good.
Hey, congratulations for your very good point. And in your opinion, what is the better next generation sequencing platform data to use in whole genome sequence selection studies?
Okay, tricky question. I might offend some people in the audience here. We're using the Illumina technology at the moment because for us it just produces so much data and so cheaply as well. And you can apply, as long as you apply good quality control, you get good results out of the Illumina platform. Now, having said that, I don't have much experience with things like solid.
Yeah, here. Hi. I was wondering, so here you're interested not only in something that is associated with a genome-wide association, but also the phase of the association is very important. So if you are saying that the allele of the marker, it's the one that has the positive additive value, then if there is a recombination in that bull that you are promoting because of the high breeding value has a recombination, has in the other sense, then all the estimates of the progeny are going to be wrong. So my question is, how much data do you have proving after how many generations that this is actually the response that you are predicting with this technology is actually what you are getting?
Yep, very good question. What we see at the moment is these associations hold up well if you predict the next generation and the generation after that. But after that, you get a rapid drop-off in the accuracy of your prediction. And it's because these associations are breaking down. And it's actually even worse if you try to predict across breeds. If you try and predict across from Holstein into Jersey, for example, the associations don't hold up. The good allele in Holsteins is usually not the good allele in Jerseys, or about 50%. So your prediction from Holsteins into something else, another breed, tends to have an accuracy of 0. So we're hoping that the sequence data can help solve this problem.
little question. I'm not sure if for animal breeders the genotype for environment interaction is important or not. For us plant breeders, this interaction is very important. How do you manage the genotype by environmental interaction with genomic selection? What is your idea about this?
Okay, so for animal breeders, it is less of a problem. I understand, my limited understanding of plant breeding is that it is a significant problem in plant breeding. And I think what you just have to have, if you're trying to predict across environments, is observations on those individuals in those different environments. So you have to have reference populations that are essentially replicated across the environments. And of course that increases your costs of applying the technology. But to predict across the environments, you just would have to have that.
Do you have listened something about genomic selection in polyploids?
Yes, just starting to try it now. And where we have tried it, or where colleagues of mine have tried it, is Atlantic salmon, which are mosaic tetraploid, so they're partly tetraploid. And the approach they took was basically to ignore the polyploidy and just predict straight off the arrays like you would in a diploid. And that worked reasonably well. But I think, especially in species like wheat, where you have A, C and D genomes, you're going to have SNPs that are specific to those genomes that let you predict the effect of those genomes ultimately. So as a first pass, you can treat it like a diploid, but to get the most out of it, you're going to have to do more. Okay. Thank you all.